Hello and welcome to the channel. In this video, it's going to be all about deuterium, and it's very suitable for beginners and deuterium enthusiasts because it's the history of deuterium and deuterium-depleted water, explained in a scientific but simple way. So what inspired me to make this video is this booklet here. This was written by Victor, who's the CEO of Litewater, and it's a really excellent little booklet. So it's his work, and I've made it into some slides and a presentation for you so that you can understand a bit more about deuterium. And I learnt a lot from this little booklet as well. If you're interested in learning how to deplete deuterium using ketogenic diets, low-carb diets, and fasting, as well as a quantum approach to deuterium and food, then check out my new Quantum Ketosis course. It's for practitioners, but also for keen lay people as well. There's a waiting list, and you can sign up now, and you'll get a little bit of information about keto diets when you sign up to get you started. Here are some interesting extracts from Victor's book on the history of deuterium. He's the CEO of Litewater, and he produces deuterium-depleted water, but also other things relating to hydrogen and deuterium, like deuterium tests, hydrogen machines. So let's have a look and see what's inside. In the first 60 seconds following the Big Bang, in the primordial plasma soup that was to become our universe, the first element was created. This element was hydrogen, made simply of one proton and one electron. The temperature was 1 billion degrees. Electrons and positrons annihilated to make photons, while protons and neutrons combined to make deuterons, a proton and a neutron pair. Almost all of the deuterons that combined to make helium. Primordial matter was thus separated into two unequal parts, 1/4 helium and 3/4 hydrogen. This process, however, was not completely thorough. As the universe cooled, a small percentage of the deuterium nuclei remained as they were, unpaired and isolated, trapped between hydrogen and helium. Most became the source of star energy along with hydrogen. The deuterium atoms that survived, the stellar furnaces, eventually combined to 2-to-1 with oxygen to create water, now found in seawater and fresh water on Earth. There are in fact about 6 drops or 300 milligrams of deuterium in each litre of water on our planet. Fast forward almost 14 billion years to when Harold C. Urey and his colleague Ferdinand G. Brickweed and George R. Murphy at Columbia University proved the existence of deuterium. Before their discovery, it was believed that hydrogen contained one proton and one electron. This rare, heavier isotope of hydrogen with an added neutron doubles the weight and has a mass of 2. Deuterium had gone undetected by physicists, perhaps because it made up only 0.014% of all hydrogen in the universe, or one heavy water molecule for every 3,300 of regular water molecules in seawater. Some physicists suspected the existence of a second isotope of hydrogen as early as 1913, and Urey was keen to prove it true once and for all. With certainty, he knew it must exist because something between the atomic weight and the mass spectrographic value of hydrogen just simply didn't add up. And with the Brickweed's help at the cryogenic laboratory at the National Bureau of Standards, they unlocked the mystery that had proved its existence. The excitement of what they discovered created a brisk back and forth to name it. Barogen, haplogen, diplogen, deuterium, and digen were all suggested. Eventually, they settled on deuterium, from the ancient Greek deuteros, meaning second. To single proton hydrogen, this makes up 99.98 of all hydrogen that they gave the name protium. In 1932, Dr. Urey won the Nobel Prize in Chemistry for the monumental discovery that would usher into the atomic age. Concentrated deuterium, i.e. heavy water, was the missing piece needed for nuclear reactors and making atomic bombs. It can be stated that because of Urey, Brickweed, and Murphy, the world would never be the same again. A third radioactive isotope, tritium, 2 neutrons, is found in 1 in every 41 million hydrogen isotopes, so it's almost non-existent. While the Nazis brought a paradigm shift in physics, much was the same with molecular biology. In 1929, ATP, the so-called believed fuel of life, was discovered. This came 39 years after the discovery of mitochondria, where ATP is created. In 1937, the Krebs cycle, also called the St. George or the citric acid cycle or the tricarboxylic acid cycle, was discovered, describing the mechanism by which ATP is produced. But we would have to wait another 60 years until deuterium's on ATP would be fully understood. Urey's mentor, Gilbert N. Lewis, professor of chemistry at Berkeley, was the first to create pure heavy water via electrolysis in 1933, shortly after Urey proved its existence, and subsequently he was the first to observe this heavy water, when frozen, would sink instead of float like normal ice water. He also observed it strongly delayed the reproduction of microbes and retarded the growth of seeds. A new era focusing on this newly discovered hydrogen isotope, deuterium, was ushered in. Soon after Professor Lewis's heavy water observation, Oscar W. Richards, a researcher at the Osborne Zoological Lab at Yale University, showed that the splitting process of sugar with yeast is 9 times slower in heavy water, or deuterium heavy water. What looked and felt like normal water was something different altogether. From 1934 to 1939, H.G. Barber and his colleagues from the pharmacological department at Yale initiated the first systematic study on the effects of heavy water on mice. Between 1933 and 1939, there were 216 published English language studies on the biological effects of deuterium, all arriving at the same observation: heavy water was an impairment on life. The experiments replacing normal water with just 30% heavy water caused bacteria, plants, and animals to die in a matter of days. More research needed to be done, but as World War II approached, heavy water became increasingly difficult to obtain for further study because of the great demand in the military sector. Biological research on deuterium stifled and gradually faded away until the 1950s. It was about the same time Francis Crick and James Watson announced the double helix structure of DNA in 1953. Genetics graduate student named Gennady D. Bredzinev at the University of Tomsk in Siberia in the Soviet Union was urged by a colleague, Boris N. Rodimov, a biophysicist, to investigate a very peculiar anomaly concerning lifespans of the Soviet population. While the average percentage of centenarians in all of the Soviet Union was less than 10 per 1 million, in certain areas of Siberia there was a striking number of centenarians, 324 per 1 million. Furthermore, most of the people of Altai and Yakutia enjoyed great health and vitality well into their old age. Knowing that these regions were uniquely provided with pristine glacial melt water from high altitudes, he was motivated to investigate this factor as a possible common denominator of the longevity of their inhabitants. Scientists focused on the possibility that some unique and unrecognized water characteristics might be involved, a hidden mystery perhaps in ancient glacial ice. The first experiments consisted of mining permafrost at a depth of 20 meters and melting the water that had lain as ice for 300 million years. In the lab, it was observed that this water stimulated cell division and slowed down aging. When the Institute could no longer pay for the extraction of this ancient ice, they evaluated Siberian snow from their own vicinity, and to their surprise, it had the same effect. The theory of deuterium-depleted water was beginning to take shape. Muhachev at the Tomsk University in 1959 to 1960 convinced his colleagues that even a small dose of deuterium distorted the chemistry of hydrogen bonding and inhibited submolecular processes. By 1960, Berdyshev had enough information to conclusively link the longevity of the Yakuts and the Altanians with the consumption of glacial meltwater. The research from Tomsk discovered that ancient ice, high-latitude mountain snow, and glacial runoff were 15 to 20% depleted in deuterium compared to what became known as standard Vienna mean ocean water, VSMOW, which is about 155.76 parts per million at the equator. As a matter of historical record, the findings were published in an agricultural journal in Omsk in 1961. No sooner had Rodimov, Muhachev, and the rest of the team discovered this rejuvenating water that a level 6 nuclear disaster occurred at the Kishtim nuclear power plant in the southern Ural Mountains, the third largest nuclear disaster in history. Berdyshev and his colleagues provided their new discovered mineral meltwater to a number of the victims, and they were saved. It wasn't until after the Soviet Union fell that the Russians declassified the disaster, as well as how deuterium-depleted water was used in medical treatment. In 1966, Rodimov and his biophysics department chair, Toroptsev, were allowed to publish their work in English for the benefit of researchers and scientists everywhere. With their groundbreaking findings in the biological role of heavy water in living organisms, they put Siberia on the map. They became the first scientists to show how water depleted in deuterium had a positive biological effect. In the mouse experiments, they observed the increase of heavy water to 3% caused offspring to have a 20% lower birth rate, 3 times smaller adult mice than control, and these mice were enabled to reproduce to a 3rd generation. In another experiment, mice consumed glacial meltwater had a greater mice consuming glacial meltwater had activity and grew faster and bigger than the control group. These experiments were repeated many times in Soviet institutions with different animals and plants. Considering that deuterium had only been discovered 30 years before, it was a monumental breakthrough. The secret of longevity had just been revealed. Coincidentally, around the same time, one of the greatest revolutions in biology was taking shape by Paul D. Boyer, a molecular biologist at UCL. He discovered that tiny protein nanomotors within the mitochondria, sitting at the end of the electron transport chain, ETC, bore the final burden for creating ATP. This protein assembly, spinning at about 900 RPM, had the structure and function of a mechanical motor, complete with a rotor, stator, and magnetic field. Boyer christened it ATP synthase, and it would be another 40 years and the turn of the millennium before deuterium's effect on ATP synthase would be uncovered. In the early 1960s, it was clear that deuterium, although a hydrogen isotope, was something different altogether, both biochemically and biophysically, being twice the mass of protium due to the addition of a neutron, which is absent in normal hydrogen. No other element has such extreme differences in mass amongst its isotopes. Nevertheless, the understanding of how deuterium functions at the cellular level was yet to be discovered. While the Russians were doing their research and making quiet breakthroughs, Americans were also hot to blaze a deuterium trail. It was 1963 when John F. Thompson of the Medical Research Division of Argonne National Laboratories in Illinois wrote the definitive 152-page treatise entitled Biological Effects of Deuterium. The work of his colleagues Joseph J. Katz and Henry Crespi reinforced the biological implications of deuterium, noting early on in Deuterated Organism Cultivation and Uses, published in 1966, that deuterium affects the shape of proteins and the replication of DNA. Laboratory mice experiments were conducted in which their normal body water was altered in the percentage of heavy water, and this yielded the following results. Laboratory mice body water was increased in concentration with heavy water or deuterium water by 30%. This proved fatal to mice in a matter of days. Laboratory mice body water was depleted in deuterium by 30%, down to 105 parts per million, resulting in significant increase in lifespans. 10 years later, in 1974, again at Argonne National Labs, British scientist T.R. Griffiths, at the Second International Conference on Stable Isotopes, proposed the theory that deuterium might be the primary cause of aging. In possible roles of deuterium and the initiation and propagation of aging and other biochemical mechanisms and processes, Griffiths states, deuterium adversely affects the shape of enzyme molecules which are involved in DNA replication. Griffiths observed that deuterium, being more electronegative than hydrogen, twice as heavy, and having different atomic binding properties than normal hydrogen, also called protium, interfered with DNA replication. When DNA repair enzymes contain deuterium in a position reserved for protium, they have the potential for participating in an error reaction thereby compromising DNA replication and repair. The following year in 1975, J.D. Gleason and I. Friedman replicated the Russian findings on plant growth, publishing the first American study on deuterium-depleted water, DDW, to increase the growth of grains. This small but significant publication in Nature magazine paved the way for a new generation of scientists to try and understand deeper functions of deuterium in the biology of living things. When the Hunza people of northern Pakistan were investigated for their increased longevity and lack of illness, it was determined that the deuterium content of their water from the glaciers of Mount Ultar was about 133 parts per million, a deviation of about 16% from 155 parts per million, which is the global standard. A 16% reduction may not seem significant. However, Griffith's theory further predicted that the adverse biological effects of deuterium is proportional to the square of the concentration, and that is the reason we now know that even a slight depletion of deuterium has a great biological effect. By the 90s, pivotal research was being done in Romania and Hungary. W. Bild and his colleagues at the Romanian University of Medicine and Pharmacology showed that mice exposed to a sublethal dose of 8.5 grays of radiation had a greater survival rate on deuterium-depleted water. Mice consuming water that was reduced to 30 parts per million deuterium had a 61% survival rate, whereas the control group exposed to radiation consumed plain tap water at 155 parts per million, and they had a survival rate of only 25%. The test group also maintained normal white blood cell and red blood cell platelet counts as compared to the control group, which didn't. The same two groups of unfortunate rodents were also infected with pneumonia, and the test group showed an intensification of immune defences not seen in the control group. The scientists concluded that mice with a lower level of deuterium in their system would benefit from less error-prone cell division and more effective repair of radiation-damaged DNA. It was proof yet again that deuterium-depleted water had some unknown and seemingly miraculous biological effects. These animal tests were carried out for the sole purpose of evaluating the effect of deuterium depletion for patients undergoing chemotherapy. This, along with the work of Hungarian Nobel Prize winner Albert St. Georgi, inspired the work of Dr. Gabor Somolai, a doctor and molecular biologist who in the early 1990s undertook the most extensive clinical trials of deuterium depletion yet completed. His data published in 1998 in the paper The Biological Effects of Deuterium Depletion and in his 2001 book Defeating Cancer, Dr. Somolai's double-blind clinical trials showed first, that deuterium-depleted water was free of any side effects, and second, the survivability of his test group was significantly better than those cancer patients in the control group who did not receive deuterium-depleted water. Dr. Somolai showed that consuming deuterium-depleted water was an excellent complementary adjuvant to conventional radiation and chemotherapy. Between October 1992 and the spring of 1999, Dr. Somolai and his team administered some 350 tons of deuterium-depleted water to approximately 1,200 patients, generating over 12,000 pages of documented records. Currently in 2019, Dr. Somolai has 22 case studies on deuterium-depleted water. His groundbreaking work put Hungary on the map as an important centre for research on deuterium depletion. Meltwater cure. After the fall of the Soviet Union, Berdyshev, now a distinguished chair of the genetics department of Taras National University in Kiev, started his new department in juventobiology. His work no longer a sensitive national security matter, he and his colleagues built 3 industrial machines to recreate glacial meltwater, able to reduce deuterium levels by 30 to 40% to about 90 to 105 parts per million. These engineering masterpieces separated deuterium out using freeze-thaw cycles, taking advantage of the fact that heavy water freezes at a slightly higher temperature. It was also publicised that people without access to deuterium-depleted water could do a similar thing at home, called the meltwater cure. The instructions called for performing repeated freeze-thaw cycles using a household freezer. By removing the water that freezes first, repeatedly, 5% reduction was claimed to be achieved. This protocol became popular in Russia and many other countries from the 90s until today. However, it was only marginally effective, as deuterium levels using this method could not be reduced significantly enough to make a major difference. By the beginning of the 21st century, it was well understood among researchers that consumption of deuterium-depleted water protected DNA from damage, but it was not yet known how. Thanks to man's insatiable desire to uncover the mysteries of life, it would only be a matter of time until this puzzle was solved. In 2006, a Russian chemist, Igor Pomyktin, and his colleagues published the study Relationship between natural concentrations of heavy water isotopes and rate of hydrogen peroxide generation by mitochondria. They were able to show that whatever mechanism allowed heavy water to damage the cells and vice versa, allowed light water or deuterium-depleted water to keep it healthy, was happening somewhere in the mitochondria. The study showed that heavy water inhibited mitochondria's ability to produce hydrogen peroxide, which acts as a messenger molecule sending signals to regulate oxidative stress. Pomyktin's work would lead to the same conclusions about deuterium's effect on ATP as was published the following year. The year 2007 marks the occasion of most monumental discovery in the short history of deuterium science. Abdullah Olgun, a medical doctor, biochemist, pharmacologist from the Department of Biochemistry and Clinical Biochemistry at Gülhane School of Medicine in Ankara, Turkey, published the paper Biological Effects of Deuterination, ATP Synthase, an Example. Devoted to the study of the mechanism that caused aging, it took Dr. Olgun an exhausting 2 years of research, as well as having to obtain another degree in medical mathematics to show for the first time how deuterium caused damage. It occurs in the last step of the electron transport chain within the mitochondria at the ATP synthase, the nanomotor. Olgun determined that roughly every 15 seconds, a bare deuteron, a proton-neutron pair, for which there is no open receptor, impinges on the fast spinning of the nanomotor, causing it to jam, stutter, and ultimately break down. Olgun further explains in the paper Deuterination and Aging, published in the same year in the Annals of the New York Academy of Science, that this is one of the key primary causes of aging. The mystery of how deuterium damaged life was finally revealed. The Nobel Prize-worthy significance of Olgun's finding may be hailed as one of the greatest discoveries of the 21st century and one of the greatest oversights in biology. At the time, only a handful were keen to take notice and register the gravity of the breakthrough. One person that got the picture right was Anton Chernopiakto, a Russian businessman, scientist and deuterium depletion enthusiast who co-authored with Pomyktin and Oxford scientists in the 215 study, Deuterium content of water increases depression susceptibility, the potential role of serotonin-related mechanism. Having embraced the importance of deuterium depletion from an early age, a lifelong quest was about to manifest. Chernopiakto, now having definitive proof of deuterium's role as a biological Trojan horse, took over the task to advance light water production beyond laboratory and research purposes and construct a factory to produce deuterium-depleted water or light water on a commercial scale. While Berdyshev in the 90s created an industrial process for reducing deuterium by 30 to 40% using a refrigerated process, it was at the turn of the millennium that the first vacuum rectification column exclusively for the production of light water or deuterium-depleted water was developed at the Institute of Fine Chemical Technologies in Moscow. Applying principles from decades of heavy water separation to the removal of light water, this was a much more efficient process, and it could remove 97% or better of the heavy water or the deuterium. In 2008, Chernopiatko, having purchased intellectual property and having taken a number of years to improve upon his technology, began the construction of a plant in the Russian countryside, commercializing the technology developed at the Institute in Moscow. A decade of prototyping, industrial engineering, hard work and luck yielded the first dedicated facility in the world to produce 90% plus deuterium-depleted water continuously. It is in 2019 the mechanisms of deuterium and how to remove it are well understood, yet there's still much more to be learned, and the awareness of this colossal discovery is still in its infancies. 4 international conferences on deuterium depletion in Budapest have provided a place for scientists to present their work. Dr. Laszlo Boros, MD and professor at the University of California, is an American scientist advancing the work in this field and perhaps the most knowledgeable biochemist in the world on the subject of deuteronomics. He co-founded the Center for Deuterium Depletion. Dr. founded the Center for Deuterium Depletion in Los Angeles, where deuterium testing and treatment protocols were first offered to the public. At the moment, there are 4 primary producers of deuterium-depleted water: Vividi in Russia, Chlorivia in Romania, Preventa in Hungary, as well as 2 plants in China. Vividi's production is offered in the United States as litewater, the exclusive and only deuterium-depleted water available in the world, where 94 to 97% of the deuterium has been removed. Victor, who's the CEO of Lightwater, and other scientists in the field believe that the ideal deuterium levels for the body are below 120 parts per million. Achieving this requires drinking 1.5 litres of 80 to 100 parts per million DDW daily for 45 to 60 days, while minimising the intake of all other beverages and maintaining a modified ketogenic diet with intermittent fasting and targeted photonic stimulation. That just means appropriate light exposure, sunlight, and minimizing blue light. Victor has personally been able to reduce his deuterium levels well below 100 parts per million, and the energetic benefits are qualitatively profound. As one would expect, when there is something too good to be true, there has to be a catch, and that is the shortage of supply. The extraction of these 6 drops of heavy water per litre is a complex, multi-level technical process requiring large-scale industrial installation of specialised distillation rectification equipment. Unfortunately, it is not a machine you can have at home. In the future, as awareness and demand increases, new production plants will come online, and even more efficient deuterium-depleted water technologies will be developed. Many countries are now actively pursuing their own interest for heavy water separation. In the 90 years from the first time hydrogen's second isotope was discovered until now, the world has changed unlike any other time in human history. We stand at the dawn of a new age of technology and the gateway to a quantum leap in biology. Thanks to the pioneers herein, radical life extension now has a powerful new ally, which is deuterium depletion and deuterium depleted water. So after reading Victor's booklet, I did decide to do another deuterium depletion, and because I'm in the States at the moment, I was able to order some light water. So you can get it in glass bottles as well. I bought more than one case because everybody in the household, apart from the children, are probably doing the depletion as well. Thank you very much for watching. I hope you found this useful and informative. If you're interested in using ketosis if you're interested in using ketosis and quantum biology to deplete your deuterium, or just to learn more about it, be sure to check out my new quantum ketosis course. It's a practitioner course, but it also comes with CPD or continuing professional development points. The link's in the description. So you need to sign up for the waiting list first, and I'll send out little tidbits of information about keto diets for you so that you can be prepared if you decide to go ahead and do the course.